It's the final year of Jung's life. He's working on man and his symbols, his final message to the public, his final chance to set the record straight. And buried in his draft, in a single throwaway line, is something that I've never seen anyone treat as the Ariadne's thread that it could be. The anima results from the minority of female genes in a man's biological makeup. Read that again. That's not poetic flourish or a metaphor. That is Jung directly linking archetypes to genes. But this is where things start to go Da Vinci Code. When Man and His Symbols comes out in 1964, that line is deleted, erased. There's not even a footnote or an editor's comment. It seems that somebody didn't want it in print. Yet the original draft, it's still there in Collected Works Volume 18. And to prove Jung really believed it, here are 4 more examples across his collected works where he restates this exact theory. Today, we're going to follow that deleted idea through the history of genetics, into the machinery of the cell, all the way up to cutting-edge systems biology. And we're going to see why, in 2025, that idea might just be key for rewiring how we understand the psyche altogether. If you like these deep dives, then get on over to the Jung Project. That's my weekly members-only podcast and private Discord server, where we go through Jung's collected works literally line by line. The first 4 episodes are out right now, and you can listen to all of them instantly. You can check out episode 1 for free on this channel. So here's the question, and we're going to plant our stake in the ground. If there really is a link between archetypes and genes, how would we even begin to go about proving it? To answer that, we have to rewind the clock back, not to 1961, the year of Jung's death, but a full 100 years earlier, and follow the story through in a way that you might not have heard before. Only that way can we see why Jung thought that archetypes came from genes, and also how close cutting-edge modern biology may be to solving the problem. And it all begins with a monk, a garden, and some peas. It's the 1860s, and Gregor Mendel, an Augustinian friar in what's now the Czech Republic, is wandering around his monastery doing his monk-like things. That's basically very deep study in all kinds of stuff, such as mathematics, all the way through to meteorology. He's doing preaching and religious things, but unlike the other monks, he has a rather strange obsession, and it's an obsession that doesn't exactly scream world-changing science. He's breeding pea plants. Green ones, yellow ones, smooth ones, wrinkled ones. And he's not just breeding them, he's counting them. He doesn't use a microscope, he has no knowledge of DNA, and the word gene isn't to be invented for the next 40 years. But all he has is a hunch that inheritance might be a little bit different to the way that people have been modeling it for the last 100 years. Up until this point, people had the idea that traits from your mother and your father would blend together, like 2 bits of paint that you mix together. But Mendel had the idea that actually, what if that's not the case? What if there is no blending? What if there are invisible packets somewhere within us or somewhere within these pea plants that were responsible for being the units of inheritance? He doesn't just dabble either. He's running thousands of controlled crossbreeding experiments and subjecting his results to statistical analysis at a time when statistics hadn't really been invented properly in the modern sense. That wasn't to come for about another 60 or so years. In total, over 8 straight years, Mendel crossbreeds 28,000 pea plants. This is where the stuff that you might vaguely remember from school comes in. He discovers from his analysis things called dominant and recessive traits. Those words, they come from Mendel. If you cross a pure breeding yellow pea with a pure breeding green pea, and all the offspring are yellow, well, that means that yellow is dominant. There's no blending between green and yellow. There's an invisible packet somewhere, and the yellow one wins out. If you then cross those yellow offspring with each other, you suddenly get a 3-to-1 split. 3 yellow for every green. The green apparently disappeared in one generation and then appeared again in the next. This is something that people say today all the time, right? Traits skip generations. He publishes his results in 1866 and presents them to his colleagues in his hometown. And none of his colleagues know what the hell he's going on about. You can find his paper, it's online and you can read it, and he fills it with all kinds of statistical analysis and also algebraic notation. And the response was a shrug, basically. No one knew what he was on about. Now, Mendel unfortunately died in obscurity in 1884. He was very popular amongst his hometown as well. It seems like he sort of gave up his more scientific ways to just go and be a really nice person. Everyone repped him around his hometown, and he got into things like tax disputes with the local authorities to do with monastery land. But that was it. The so-called father of genetics, who we all learn about in school, was an obsessed, brilliant monk who nobody took seriously. But in the year 1900, that's 16 years later, 3 superb botanists, Hugo de Vries, Carl Correns, and Erich von Tschermak, rediscover Mendel's work. There was a big controversy at the time as well. Hugo de Vries, one of the most popular scientists across Europe, he seemed to have independently repeated Mendel's experiments and, rather suspiciously, used the terms dominant and recessive, exactly word for word, to describe the same results that Mendel got. Carl Correns called him out in print, and all 3 of them, who had done very, very similar work, pushed Mendel's work as hard as they possibly could across Europe. Overnight, Mendel is suddenly recognized as the man who cracked the rules of inheritance. But what nobody could tell you yet was where those units lived. This is where the story begins to accelerate. Thomas Hunt Morgan, an American geneticist, enters the scene with a lab full of fruit flies. If you've never bred fruit flies before, this may come as a surprise. They're perfect for this kind of detective work. They've got short lifespans, they breed like absolute mad, and their traits will show up in neat, trackable ways. So Morgan starts crossbreeding flies with different eye colors and wing shapes, and the results don't just match Mendel's ratios. They show a location. The units live on chromosomes, physical structures inside the cell nucleus that are lined up in pairs, like matched books on a shelf. And just like books, the order matters. If you break the sequence in these chromosomes, if you swap a page by metaphor, then the trait will change. By the 1920s, our picture sharpens. Women have 2 X chromosomes, and blokes have 1 X and a way, way, way smaller Y chromosome. This is the biology, obviously, that you and I grew up with, and it's also the biology that Jung grew up with. But the difference is, for you and I, we grew up learning about DNA. In Jung's day, they knew that chromosomes were where the units of inheritance were. We could map it, we could look at it under a microscope, we knew it pretty solidly, but no one could tell you what those units were actually made of. Inside the nucleus, under a microscope, chromosomes look like weird coiled things. Some of the time they're in those nice, cute X shapes, the karyotype it's called, that most people are familiar with. Today we know that a chromosome is just one long molecule of DNA, just very densely packed together. But circa 1900, we had absolutely no clue. If we rewind the clock again to the 1860s, a Swiss chemist called Friedrich Miescher, not Friedrich Nietzsche, but Miescher, uncovers something he calls nuclein. He takes pus-soaked bandages and does chemical experiments on them, and later on he took salmon sperm, of all things, mostly because they're basically just nuclei on their own, and filtered out a substance that all he could call was nuclein, that is, a strange substance within the nucleus. Fast forward 60 years, and this strange substance gets a brand new name. In the 1920s, it's renamed DNA, or deoxyribonucleic acid. We know what the chemical structure is at this point. What today we learn of as the A, T, C's and G's, but without any of the detail. We just know roughly what types of atom are present in this nuclein. But the thing with this is nobody suspected that DNA would be where inheritance is carried through the generations. In fact, for the vast majority of scientists, their money was on protein. Proteins are complex, they're versatile, and they can form basically endless combinations. They're far more glamorous than the bland, repeating pattern within DNA, or DNA just looked a bit too boring. Then came a 1-2 punch in the history of science. In the 1940s, Avery, MacLeod, and McCarthy show that DNA can carry heritable traits in bacteria. Then in 1952, Hershey and Chase seal the deal with their famous blender experiment. We all know today that viruses will inject their DNA into a cell so that that cell will then make more copies of the virus, making us all unwell. That's exactly what Hershey and Chase proved. It wasn't protein being injected in, it was DNA. So now at this point, 1952, the entire scientific community, more or less, now agrees that it's DNA, which is where Mendel's invisible units live. And then the most famous moment of all, in 1953, James Watson and Francis Crick, using X-ray diffraction images from Rosalind Franklin, uncover the beautiful, now a brand across the world, obviously, double helix shape of DNA. Two intertwined strands, base paired like rungs on a twisted ladder. The whole molecule is directional, anti-parallel, and elegant. It's the kind of structure that makes you feel like you've just been handed the master key to all of nature. Each rung, A with T and C with G, are information. The order that those letters go in along the chromosome is the script, literally a script for building an organism. The twist of the helix, too, that's not just for looks. It allows the molecule to be copied with uncanny precision every single time the cell divides. Up until this point, the word gene was used in a rather linguistic way. It did not have a molecular basis at all. It was just a term for one of Mendel's units living somewhere on a chromosome. But from Watson and Crick's work, we now knew that it was not a metaphor. They're sequences, physical stretches of DNA coding for something real, something material, and something that you could, in principle, read like a book. The hunt was now on to crack the code. So this was the question. How do those four letters, A, T, C, and G, become or somehow create you, all the proteins within you that make you unique? Well, in 1961, again, that's the year of Jung's death, Crick and Brenner and their colleagues run a series of brilliant genetic hacks on viruses and bacteria. They show that the code is written in triplets. That's 3 bases corresponding to, let's say, 1 word, with each word specifying something called an amino acid. Suddenly, DNA has a language based on the number 3, and a language that's exactly the same in every single living thing. By 1966, Nirenberg and Correns had finished the job. We'd now got the entire codon table, that is, the Rosetta Stone for translating DNA's language into what proteins it's going to create. It's at this point that Jung's female genes line, it starts to come apart, because there's no such thing as a male gene or a female gene. It's all context dependent. It all comes down to how a cell reads and interprets the language within DNA. By 1961, we know that men do not carry a little sprinkling of female genes. Blokes have a full X chromosome, after all. We might say, well, yes, but women have 2 X chromosomes. That's double the X load, right? No, according to Mary Lyons at the time, brand new X-inactivation theory, we know that in female cells, one of those X's, weirdly enough, is shut down. So that means that the active X dosage is roughly the same in both men and women. That's an example of epigenetics in the wild. That's a change from the output you get from the genome without changing the sequence of DNA itself. We're going to talk much more about epigenetics in just a moment. There is a male gene, it's called SRY, and its function is to masculinize a fetus into a proper bloke. But there is no female gene. Perhaps this is why John Freeman, that's Jung's publisher, or Marie-Louise von Franz looked at Jung's draft and thought, "Nein, this is silly." But if we remove the word female, then Jung's stance is still crystal clear. Archetypes come from genes. From our story so far, it's looking very likely that there is no such thing as an archetype gene. We hear this all the time, don't we? There's a gene for being fat. There's a gene for being thin, or a gene for being tall, or a gene for a certain whatever. None of this is true. And if we're going to really take this seriously, something like an archetype is going to be very, very complicated. Anything within the psyche is going to be very complicated. So it's highly likely that the link between archetype and gene is not one-to-one. It's going to be in how the same universal code gets switched on, silenced, or rewired. This is where we're going to go full professor. We're going to move all the way up to 2025 now and do a very comprehensive walkthrough through the entirety of gene regulation. If you don't have any biology training, do not worry. I'm going to do my very best to explain this in a simple fashion, but that doesn't skip out on detail. I despise summary videos. You always come away wondering what if and what if. So we're not going to do that. We're going to look to see where these instructions, where Mendel's units actually truly live. And the answer is, it's not in the genome itself. It's actually in the entire cellular system. So grab yourself a beer, or if that's not your style, a coffee or something more boring, and let's go. We need to debunk a nonsense myth straight off the bat. DNA is not a blueprint. It's a library, but a library in which most of the books are behind the restricted section, just like in Harry Potter. So here we go. Let's begin with the genome. So what does that term even mean, genome? It's literally just the sum total of all of your chromosomes, plus the tiny bit of DNA that's in your mitochondria. That's literally it. And it's about 3 billion base pairs long. Those A, T, C, and G letters, they form genes. But only a small fraction of your DNA is actually protein coding. And until about 20 years ago, we thought that, therefore, only 1 to 2% of your entire genome was useful for anything. The rest was just noise. Junk DNA, it was called. We know today that the rest is regulatory sequences or stuff like structural scaffolding and even weird stuff, this is real, like viral insertions. They're called transposons. And a ton of stuff that we're still trying to figure out, such as the sequences that code for long non-coding RNA. This was something that I worked on myself in my PhD research before Jung dragged me off course. DNA doesn't just float loose either. It's actually wrapped around proteins called histones, forming something called chromatin. Now, this packaging is really dense because if you stretched it out inside every single one of your cells, your DNA would be 2 meters long. I'm almost 2 meters tall, so that would be like taking me, smushing me down, and putting me into every single one of your cells. It's a rather uncomfortable thought. So your genetic information, that's the stuff that makes you, all of your proteins, is locked in this dense mass. How the hell does the cell know how to read it? Well, checkpoint 1. How open is this chromatin? Seriously, chromatin can be either locked up tight like a hermit or have its arms wide open like a bloke who loves to cuddle. If the histone proteins are acetylated, this usually opens the chromatin. And if the DNA itself is methylated, often a gene is switched off. Those chemical tags are epigenetic marks. That's basically what epigenetics means. How can you modify what comes out of the genome without changing the genome itself? It's a term, obviously, that's been mythologized all over the internet and turned into all kinds of strange things, like most science terms. But that's basically the core of epigenetics. And I know that I'm name dropping a lot of mechanisms here, but the main thing to take away is before we even get to gene expression, there's something going on. There's a checkpoint beforehand that decides which part of the genome the cell is going to read. Very, very tightly regulated and very complicated. That means that in all likelihood, we are not going to be born with all archetypes or even all archetypes fully matured within our psyche. Instead, it's much more likely that they are going to be slowly released across our lifespan. Or if a certain stimulus comes up in the environment, gene expression is triggered off to bring that archetypal material online, whereas perhaps beforehand, it wasn't needed. That's the beauty of this checkpoint system with the genome. Now, if the chromatin is open and the cell actually wants a particular gene on, it has to copy it into messenger RNA or mRNA. This process is called transcription, and it's not casual. It's a full-on choreographed dance. You've got promoters. So these are short stretches of DNA that act like a loading bay, where all the transcription kit gets assembled, ready to go. Let's transcribe this gene. Then there are enhancers and silencers, and they can be thousands of bases away, but they still dial the volume up or down on how much of that gene you want to be expressed. And then you get transcription factors. These are proteins that grab onto those promoters and enhancers, and they flip the switch to on or off. And finally, the star of the show, this is a rather crude representation, I'm sorry, but RNA polymerase II, the molecular machine that physically builds the RNA strand from the DNA template. You can watch it go. Here's the thing with this too. You often need multiple transcription factors to show up either at the same time or in a very specific order, or else this process will not happen. The gig will be cancelled. And remember, this is a weird thing. Each of these transcription factors, being proteins, they were once genes in informational form. So that means if you want your one gene that we're studying, say at the moment, to be switched on and working, we have to really look at everything that's going to switch that on and what switched them on and then what switched them on in this massive complicated system. So you can see how deeply regulated these dynamics are. Once the mRNA has been made, it's usually considered quite a bit of a mess. It's full of something called introns. Instead of being useful, well, they probably don't really do anything. So the cell needs to edit them out before that mRNA can do anything useful at all. And this edit is done by a molecular monster, which I can't represent in Adobe After Effects, called the spliceosome, which snips out those introns and stitches the exons, they're the useful bits of the gene, together. And this is where it gets really, really clever. So the same gene doesn't have to be spliced the same way every single time. This is a process called alternative splicing. One gene could be cut and reassembled into different scripts, making entirely different proteins. Now, this is why our 20,000 genes in our genome can churn out well over 100,000 distinct proteins. It's like you're getting 10 different films out of the same reel of footage just by changing the edit. Once the mRNA is properly edited, it leaves the nucleus, and heads for a ribosome. Again, I cannot represent this in Adobe After Effects, so this is pretty close, to be fair. This is the molecular machine that builds stuff. The ribosome will take the mRNA 3 bases at a time. Remember, that's Crick's triplet code. And each triplet codes for a specific amino acid. Now we get another molecule called tRNA. These act as couriers. So each one carries an amino acid on it, one side, and on the other side, it's got an anticodon that matches the mRNA's code. So they slot in literally like puzzle pieces. The chain of amino acids grows as the ribosome moves along the mRNA, and that chain of amino acids folds into shape, and you've got a protein. Job done. Finished. Actually, no, not job finished. You still get something called post-translational modifications. This is when the cell looks at this lump of protein made out of amino acids and goes, you're not fit for purpose yet. We're going to process you further. So you might get a phosphate group that's stuck on it, or a ubiquitin tag, or a glycosylation. That's a chain of sugars stuck onto it, depending on what function that protein is destined to have. This is the part where things begin to fry our brains a little bit, because each of those steps that we've just walked through, so that chromatin remodeling, transcription, splicing, translation, post-translational modification, all of these are subject to what's called gene regulatory networks, or GRNs. So that means that you can mathematically model tons of things going on within this massive system, things like cascades, or things going up and down, and how the nodes in this network, each little molecule, interacts with each other. In other words, these things aren't necessarily linear cause and effect. A causes B. A classic example would be a hormone. A hormone, or smug, it swims into the cell and it might smack 20 different transcription factors all at the same time. So you've got a regulatory network. DNA is obviously stated to be the blueprint, which we debunked a moment ago. Really, it sort of is, but it becomes a chicken and the egg scenario. You are born with your genome, but really that genome is born into a cellular context. When the sperm meets the egg, there's all kinds of stuff going on that's not just the DNA. So the DNA may well be, well, it is extremely important, but it's not the be-all, end-all. It's the entire system, without a doubt, that's really engaging in what could be called a living symphony. Literally, the DNA is like the sheet music, but this cell is like a 100,000 piece orchestra, all micro-tuning their instruments at the exact same time, depending on what song, very, very subtly different perhaps, that they want out of the genome. So we're going to look closer at epigenetics. What does this term even mean? Epigenetics? All epi means is above. So technically, the word just means any process taking place above, or really after, the genome. So this entire process that we've seen so far, all these different steps, if you've got regulation above the genome, we could classify that broadly as an epigenetic effect. So if we think back to Jung's day, it was very common, at least in the last 10 years of his life, for people to wonder whether or not things were one gene, that'd be one Mendelian unit, the things that Mendel looked at, to one particular trait. For some things, yes, this is the case, but for the vast majority of the time, no. And also in practice, never is this really the case. Because at any step along all of those different steps we've just gone through, you can have a sense of regulation. I'm going to cover a few examples of this because these are bloody brilliant. This first one is the relatively famous Agouti mouse. So if you feed a mother mouse a diet rich in methyl groups, have you ever had a diet rich in methyl groups? Then her pups will literally change color and body shape. They'll go from fat yellow fur balls to lean brown ones without altering the DNA sequence. They've got exactly the same genome, exactly the same. Not mother to child, obviously, but there's been no damage or mutation to the genome whatsoever. How strange is that? I think Mendel in particular would have looked at that and gone, colour? That's my ballpark. One that's more famous is what's called the Dutch hunger winter. So there were kids that were conceived during a famine, and they showed altered methylation patterns, that's an epigenetic marker, 60 years later. The environment left a fingerprint on the genome's margins. And this is where the Jungian light bulb, I feel, should start to go off. With epigenetics, we have a means of plausibly understanding how an archetypal potential within the genome isn't just a metaphor, but can actually be prescribed all the different things and properties that Jung prescribes to the archetype. For example, the stages of life. Puberty is the most obvious one. All of us go through puberty, or the vast majority of us anyway. And this is a genomic process. Under the influence of hormones acting on transcription factors, you get huge cascading changes in gene transcription. And obviously you have enormous changes in psychology and enormous changes in archetypes around this period of time. We don't have to lock it into puberty either. We can extend this right out to, let's say, the second half of life. Jung famously pointed out that around the age 35, if someone had not yet engaged with their individuation journey, then they're likely to have a midlife crisis. We can understand this, again, as cascading constant changes, perhaps more that biologists know today, regarding these archetypal potentials. And all of it can be plausibly explained with epigenetics. Another example, and this is one that Jungians bring up all the time, as would a lot of psychologists. Well, what is the role of experience in all of this? If an archetype is just an innate pattern, is there any room for, let's say, attachment bonding to a specific caregiver, as a classic example? That's not just something that's innate. It requires a very specific and individual external stimulus, something to imprint onto. Yes, absolutely, because the environment and our experience is an enormous player in our epigenetic profile. And therefore, you get nature and nurture working together. Nature is nurture and vice versa. This is completely coherent with Jung's view. Perhaps most exciting of all is the burgeoning field of epigenetic inheritance. Now, this is a subject, again, which some people take and go complete wacko mode with it. So we have to be based in what the data actually says. So for the longest time, we thought that epigenetic markers were wiped when we moved from one generation to the next. But from some experiments, most of which are done in utero, in toxicology environments, we know that this is not necessarily true all the time. There's a wonderful, brilliant study that came out a while back showing mice, and how mice obviously have an innate fear of certain things. Strangely enough, if you put cat hair or fox hair next to a mouse, they'll instinctively run away from it, as if they've got a kind of ancestral memory. But if you pair that fox or cat hair with a certain smell, a smell that the mouse has never been exposed to, then when you then show that smell on its own, the mouse will get scared and run away because of the association with the fox or cat hair. All fine, all predictable. But if they're male mice specifically, that fear, strangely enough, can be passed on to the next generation, even though that next generation have never been exposed to that smell before. And the evidence for this is certain epigenetic markers living within and not being wiped away in the sperm cells of that father mouse. Now, if we think about family constellations or family complexes or things of the kind, this is still extremely early days, but it's potentially possible. I also want to caution against the idea, because I'm feeling like a nice person today, against the idea of intergenerational trauma. I've seen quite a few people over the years take epigenetic inheritance and use it to hurt themselves, or to think to themselves that they have this 1,000-year-old or several generation long trauma within them. I don't think that's fair for people to do that to themselves. That's not what the data says. Instead, we should treat this as something that's really genuinely exciting. What from our ancestors might be within us in a positive sense. I think it's really cool. So if Jung said in 1961 that archetypes arise from genes, this is the 2025 upgrade. Archetypal readiness sits in gene networks, and epigenetics decides when, where, and how hard those networks fire inside a lifetime, and sometimes, maybe, across lifetimes. That was a hell of a journey, eh? We're going to get back to Jung now. So since his passing, the terms Jung and biology, they haven't really been used, at least favorably, in the same sentence, to say the least. There are lots of reasons for this, which we won't get into here. But there has been a smaller, parallel, but still very much accepted tradition within Jungian psychology that's taken the integration of Jung and biology extremely seriously. We can talk about figures like Adolf Portman if we want to. I do want to name drop him. He was a contemporary of Jung who looked at Jung and Konrad Lorenz and the ethological side of things. But really, the real grandfather of this tradition is the late, great Dr. Anthony Stevens, who championed the idea of archetypes existing within the genome, also within the brain. He had ideas as to where in the brain they might be. And his grand synthesis from 1982, Archetype: A Natural History of the Self, which I think, yes, visible on my shelf, is a masterpiece from start to finish and a true monolithic work within Jungian psychology. There have been others as well who have worked on this problem. I'd like to name drop a few here. So John Ryan Huell, and two guys working in code biology, Major and Vidor. I'll drop papers from all three of those in the description down below. All of them are worth a deep study. But in my personal view, I think it's fairly obvious, actually, the real flag bearer for this Jungian biology tradition today is Dr. Erik Goodwyn. To the best of my knowledge, Goodwyn is the only serious scholar and clinician who's not just taking Jungian biology seriously, but is totally unafraid to go straight into the molecular biology, into the neuroscience, and even into the philosophy and the consciousness studies side of things without drifting into metaphor and without drifting into pseudoscience. If Jung's archetypes from genes comment is the ignition key, then Goodwyn is the one at the moment who's actually driving that car. So we're going to look at some of Goodwyn's contributions here because they're very important. And for anyone out there who wants to work in this particular field professionally, or I imagine for a lot of you as an autodidact, Goodwyn's someone to seriously study. So biology has many myths. Strangely enough, some things in biology become almost pieces of the culture. The famous double helix shape of DNA, or Charles Darwin being some kind of absolute moral saint championing science, which wasn't true. And one of them, which persists to the modern day, and I think I know the reasons why for this, to be fair, is something called the impoverished genome argument. It's an idea that was popular about 20 years ago, before this explosion of epigenetics really took off, where we thought that the genome contained 20,000 genes. That's not a wrong statement necessarily. It's more nuanced than that. We can argue over the number, but that's the sentiment. Now, most modern Jungians that I've seen will still cling to that argument, and not out of malice or out of ignorance, but simply because it's not their specialism, to be totally fair. So Goodwyn's response to this idea of the impoverished genome argument is to say, okay, you guys, or a lot of you guys, might say that there are not enough genes within our genome to encode the full bandwidth of archetypal potential that Jung spoke of. But actually, if we really look at the biology properly, if we look at the epigenetics, if we look at gene regulatory networks, in particular, he highlights the lac operon in E. coli bacteria. Actually, yes, it's not just 20,000 genes. You can have so much more potential to come out of those 20,000 genes. And his reasoning is basically extremely similar to what we've already laid out and walked through in this video. You already know those different network steps. It's not just genome to reality. It's genome through massive regulatory algorithm that multiplies its potential into reality. Let's have a further look at this lac operon, which all university students in biology will study, but perhaps not seen in this context of archetypes. So this is just a tiny gene network, as we say in E. coli, that controls how the cell digests lactose. Now, it's literally only got a handful of genes, but the way that they're wired together means that the whole system can sense the environment, decide whether lactose is present or not, and then either switch on or shut down the machinery to break it down. This is just a few genes in a microbe, and yet it's already showing decision-making, feedback, and adaptability. Now scale this up to the human brain, and it's obviously fairly easy to see that the amount of potential decision-making and learning and feedback potential that we can have is absolutely huge. Neuroscience is massively on board with this. But this is another thing that Goodwyn has spoken on quite a lot, is this real myth of nature versus nurture. Could you have an archetype that's purely nature? Yeah, sure, but then you lose the learning component, right? But then if it's purely just learning, say a more object relations style approach, you lose the inbuilt nature side of things. And this is kind of a thing that we're raised with, isn't it? Is it nature? Is it nurture for all kinds of different traits that we might have? But as we said earlier, and as Goodwyn has pointed out many times, these things aren't opposites. We know with neuroscience and biology, they're not opposites. You have an inbuilt potential, and how that potential is released depends on your experiences and the environment. As I say, Goodwyn has gone much further than this as well. In many papers, he's moved off into the field of code biology as well and made some great contributions over there. So if you want to take this subject seriously and you want to get up to speed really quickly, go check out his papers. Just when you thought that the story might end at, yeah, mate, it's these complicated networks all the way down, I think we're going to pivot now into a subject that's very near and dear to my heart, quantum biology. Could there be a link between quantum biology and archetypes? I think there could be. So if we're going to tell this story, let's rewind the clock back to an example I'm not sure many people are aware of, to be fair. The year's 1963, and there's a Swedish physicist called Per-Olof Lodin. I'm not quite sure if I'm pronouncing that correctly. My Swedish is not particularly good. But anyway, he was a brilliant guy who had a very strange hypothesis. So in your DNA, A will bond with T and C will bond with G on the two strands that make up your double helix. And that bond between them is called a hydrogen bond. The hydrogen bond is not a normal chemical bond, though. It's actually a link between a proton and a lone pair of electrons. We're immediately in quantum world right now, albeit sort of at the classical scale, or at least in the way that we want to study these things. So Lodin had a strange hypothesis, and he said, hang on, lads, what if the protons in those bonds could quantum tunnel to the other side without the bonds breaking? Now, if you can, you'd get something called a tautomeric shift. So the base will momentarily flip into an alternative form. And when that happens during replication, it can lead to mispairing, so a spontaneous mutation. So Lodin's hypothesis was quantum biology might cause DNA mutation. The potential implications for this for all kinds of things, least of all evolution, is enormous. For decades, this sat as a provisional but provocative and highly interesting hypothesis. But then in 2022, the lab of the famous physicist Jim Al-Khalili at Surrey University proved mathematically that yes, it is extremely likely that double proton transfer, this special kind of quantum tunneling in DNA, could really be possible. And I was so excited to see this because in 2017, for my BSc dissertation, I worked on this story at Surrey University under Professor John Joe McFadden. Now, you may be wondering, are there any quantum biological mechanisms that directly link to Jung's archetype? If we take his original definition from 1919, an archetype is essentially the inbuilt form that structures our perception and apperception. Is there anything of this kind in the quantum biology world? Yes. Take the European robin, which can quite literally see the Earth's magnetic field. It has a protein called cryptochrome in its eyes, which contains electrons in a quantum entangled state. When the bird changes the angle of its head, the configuration of that entanglement shifts slightly, giving it quite literally a superimposed compass over the world, so that it always knows where to migrate to. This is quantum biology providing the form that structures perception, exactly the definition of Jung's archetype. Quantum biology is still in its infancy, which is a good thing, because it means there's many more exciting discoveries to be made. But it does seem that many more mechanisms than we previously thought, say 20, 30 years ago, do have a quantum aspect. An example that I'm so happy that, in my personal view, has now been confirmed experimentally is Professor Stuart Hameroff's ORCH part of the famous ORCH-OR. Hameroff's hypothesis was that microtubules in neurons can resonate backwards and forwards at the frequency of terahertz in a quantum entangled superpositioned state. In 2022, this hypothesis was essentially confirmed experimentally in the lab. Unfortunately, though, the OR part, objective reduction, that's the part from Sir Roger Penrose, this new way of modelling, perhaps, how the wave function collapses in a non-observer dependent manner, that's still not yet confirmed. If ever it was, it means that we need to go back to my Jung-Pauli video and really look at Jung and Pauli's hypothesis in more detail, because that obviously relies, at least in its current form, on the Copenhagen interpretation of quantum mechanics. It would be very exciting if Penrose turns out to be right, however. Speaking of Jung and Pauli, we've covered the biological aspect of archetypes in this video so far. But there's another aspect, dating from their collaboration in the 1950s, where an archetype was stated by Jung to be nothing but the probability of psychic events. We explored that statement together in a lot of detail in my Jung-Pauli video, but essentially what it means is perhaps an archetype is not just a biological phenomena, but is inside and outside, psyche and soma, or idealistic and materialistic, whatever words that you want to use, with synchronicity given as a core piece of evidence for this. And if Jung and Pauli are right, it would mean that the archetype is essentially synonymous with that probabilistic aspect of quantum mechanics. So does epigenetics have anything to say at all about a probabilistic element of quantum mechanics? Or indeed about a kind of formal cause or a universal shaping form that perhaps is present within biology? Yes, and also no, but there's only one person in the world that I'm aware of who's uncovered genuinely convincing evidence of something roughly resembling this within biology. And yes, before you say it, because I've had so many comments mentioning this guy's name, and rightfully so, he's brilliant and should win the Nobel Prize, Michael Levin. Dr. Levin's work is equal parts important and obscenely exciting. But something I've not seen too many people talk about when discussing his work is that technically it's epigenetics. Remember our definition from earlier, anything that takes place above the genome to modify the information flow coming out of it that doesn't touch or change the genome code itself, that's epigenetics. Levin has worked on how cells use bioelectric signals, that's literal voltage patterns across the cell membrane, to coordinate growth and regeneration and even large-scale body plans. He's shown that you can reprogram a frog embryo to grow an eye on its back, or a tail where the head should be, just by flipping those electrical circuits, and these things stay. The genome isn't being modified, but the information coming out of it certainly is in a stable fashion, and not in the usual mechanisms. He's not looking at specific molecules within these cells. He's looking at how a voltage membrane profile can translate into an enormous orchestrated, what we could call philosophically, formal cause. He's also worked to create something called xenobots. These are tiny, equal parts core, equal parts abomination cellular machines. By grafting frog cells together from different organs within a frog, he's found that these tiny clusters of cells seem to have a weird kind of emergent agency. They can do really strange things that should not be programmed into these cells, such as, weirdly enough, despite not having eyes, maneuvering its way around a maze without bumping into the side. Understandably, Levin's work has attracted an enormous amount of attention in recent years. Chances are you've not just heard of Levin, but you've watched a lot of his videos. He's very popular on YouTube. I do see quite a lot of people take some of his stuff and run with it in a direction that I doubt he'd want it to be taken in. I've heard things said many times, such as the genome isn't important, or the genome's not the be-all, end-all, in almost a dismissive type of way. But Levin's work is epigenetic. It just seems to show that there's something else going on that's, well, not in the molecules. It's also not outside the molecules. There's just some kind of agential or consciousness-based or something that goes on when we stimulate or interact with cells in a certain way that appears to be built in. Levin's hypothesis has been to postulate the concept of a Platonic space. That is, quite literally, patterns that structure our cognition are just as fundamental, built into the universe, as other factors. So instead of evolution producing things like our cognition, our intelligence in the William James style of that word, our ability to solve problems or think creatively, really, these things and these capabilities are already built into the universe in some capacity. So evolution doesn't produce it, evolution further embodies it. Personally, I really feel that he's onto something, but personally, going with a full Plato-style stance is technically an ontological preference, rather than stating what the data that he's gathered necessarily points towards. I'll give one alternative, and that is, rather than a Platonic space, why couldn't this be more Unus Mundus based? If that's the case, then it could very well be that Dr. Levin has found the first piece of concrete evidence for the Unus Mundus. Rather poetically, Pauli himself, Wolfgang Pauli, looked to the future at biochemistry specifically to uncover more evidence of the Unus Mundus. And quite possibly, Pauli was right. Levin's work is beyond superb. He is one of the most important biologists of his generation. Massive, deep respect for the work that he has done. And that takes us right back to Jung in 1961. He's old, ill, but still writing. He didn't know about mRNA, transcription, translation, Levin's work, quantum biology, the lac operon, any of it. But he had a hunch and a conviction that archetypes arise from genes. But here we are 64 years later, and the future looks brighter and more exciting than ever. One thing is crystal clear. The psyche and biology are very closely intertwined. Jung died with his work unfinished, but there are people today continuing it on in various ways. And if you have a calling to join that tradition, nothing and no one can stop you. My new Discord community already consists of people from a wide range of different fields: psychology, biology, physics, mathematics, and computer science. If you'd like to join them and myself engaging in serious scholarly research, real-time individuation, against the backdrop of a vibe that feels like a big virtual pub where everyone is always having a wonderful time, then join the Jung Project. It would genuinely be wonderful to meet you, to hear your ideas, and to see how we could potentially work together and collaborate on pushing this field forward. That's the whole point of the Jung Project, is to take these ideas so seriously that we build things together. That's how I believe things move forward. In the meantime, check out Dr. Erik Goodwyn's channel, linked down below. Go look up Michael Levin's videos, because if you haven't seen these things before, that's going to be a rabbit hole that's going to consume you for probably months on end. And stay tuned for the next video. It's another big one planned. I think you're going to really like it. Thank you very much for indulging me over the last 50 minutes or so. I'll speak with you again very soon.